Okay, so I started not really dealing with molecular connectivity because it's something, of course, related to the path you see in a different course, but I deal with relative absolute measures in molecular imaging. So it's a different way to handle the PET data, also probably the most common use that is done today. So I have to move it to the second one. It's clicking is not possible. Okay, yes, I don't have any conflict of interest. So PET imaging, we already have seen what PET imaging is. What do we need? We need a scanner as a fMRI. So when we deal with a PET, we deal with a narrow image stuff. And it's actually the same with fMRI, with diffusion stuff and so on. We need a scanner. A scanner able to detect what? Now we want to detect the emissions from the annihilation of a positron emission radionuclide, so a substance, an isotope that is injected inside the body. So we need, what I want to say that with MR we need a scanner with different type of sequences to derive different type of images with type of information. So now we need a scanner and we need a different type of not sequences but tracers, okay? Because it's based on the tracer, the interpretation, the physiological information we can derive. So I will use the word tracer. What is a tracer is? It's a substance. If you want to investigate, for instance, the glucose, the behavior of the glucose inside our brain, so you can just decide to use the glucose itself. But of course, it's not detectable by MR itself. We can do something, but it's not directly. And also, we can detect directly the normal natural glucose by using PET. We need to transform this substance in radioactive substance. How to do this? We already have seen we have the radioisotopes. For instance, one is the carbon-11. So from a principal point of view, easy to say, I think is a little bit harder to do. We can remove the natural carbons from the glucose and replace with the carbon-11. So we have still glucose, but now it's detectable by the PET scanner. So we can repeat, we can do essentially with every type of substance, because we have dealing with organic substance, with every type of substance that we want to investigate with PET. So a tracer, PET tracer, is something that is able to be detected by the scanner, so it's radioactive. But also, with tracer means that the amount of tracer is picograms, nanograms, okay? So I cannot just use 1 kilo of glucose, because in this case, I'm not going to investigate exactly the behavior of glucose, but the action of a glucose on the glucose isolate system. Or, for instance, the DOPA. I can inject grams of DOPA because immediately we have a response of the behavior from the person. So I need to use a small amount. A small amount is usable to be detected, but not able to perturb the system. The system is a steady state, as it is. Okay, so how many tracers we have? From principal point of view, each substance could be visualized by tracer, by PET something, but of course we have just a small amount of possibilities due to the possibility to produce the tracer itself by using cyclotron. In any case, we have more than 60 tracers available, so more than 60 more than a possibility to investigate different molecular behavior inside the brain, also outside the brain, but in particular in the brain. So for instance, one of the oldest is the water. In this case, it's not marked with a C11, but due to the fact that sometimes it's marked with another isotope, O15. Protein density is one of the last tracer released by Yale University. Is UCBJ, is based on the C11 as the isotope. Protein synthesis, so the rate, the velocity to produce at each voxel, the X voxel, the proteins. Serotonergic transporters we can investigate by using DASB, and glucometasmine, that is another classic PET trace is FDG. A longer list you can find there in this paper. Of course, more or less we have 1 or 2 new really interesting PET tracer each year. So now we have the scanner, we have selected the tracer. What we can do, we understood, we can act in 2 different ways. We can acquire just only a static PET image, or we can deal with a dynamic version. So this is exactly the same. The trace in this case is the synaptic density. The person is exactly the same. So we can compare 2 different images. The first one is a static, is acquired after, I don't remember exactly, after 20 minutes after injection. And so we have this that is provided as in kilobecquerel over millimeter. The original PET image static, the signal is in PET has, different from MR, has a unit of measurement. It's a kilobecquerel over millimeter. And we can, which is the interpretation of this static on the left PET image. So higher is value. We are dealing with synaptic density. Higher is the synaptic density. This is correct. Is you know, what we wanted to derive from this static image. This is a question. We have to be sure about the interpretability of the interpretation of this concentration, because it's a concentration, it's a kilobecquerel over millimeter, okay? It's just only a picture of a dynamic system that is evolving from the injection of a tracer till the end. So, or we can acquire a dynamic image, and I'm sure that I will deal. Okay, so exactly the same tracer, the same subject, but here we have more images. The possibility to follow the injection, the behavior of the tracer from the beginning, time 0, injection time, till the end, and you see the tracer coming inside, reaching the highest value, and going down because it's going outside the system, okay? Which is the best frame, which is the best of several frames that we have seen here to translate the kilobecquerel over millimeter in physiological information, okay? This is the question when we have every time to ask us when we deal with static compared to dynamic acquisition. So trying to go. Okay, static, we already seen. The question is, I inject the tracer, a small amount of dose and so on, at time 0, wait time, and acquire just only 1 image and a specific time window. Could be at the end, could be in the middle, depends on the tracer, also this one. So I have 1 image, and that's all. Good. I want to compare, normally I don't want to work at a single subject level. Normally I want to compare also different types of subjects. So the best is to translate the kilobecquerel over millimeter in some standard values. Why? Because of course the dose can be different from a person to another one. So the first step is to translate kilobecquerel in SUVr, so a standardized uptake value. Uptake because it's something that is all style version related to FDG. In any case, a standardized uptake value. It's just only the ratio between the radioactivity concentration in each voxel, by kilobecquerel over millimeter, by the dose, injected dose, that normally is provided in megabecquerel, divided again from another scaling factor that could be the body weight of a person, or sometimes the body surface area in Bmax, more frequently is used body weight. Okay, now, well, we have translated, we can compare the different type of static images throughout all the subjects. Fine. And this way to proceed has, of course, advantages and disadvantages. Advantages is very simple to acquire a static image. Of course, it's easy to calculate as it is. You have just only 1 value for each voxel, easy to handle. And And also is a standard approach of a clinical studies, so is most frequently we will have in touch with PET data, with PET images, static PET images. Is less frequently to have a dynamic PET acquisition. Why? Because it requires a longer time, and in a case, for clinical purposes, have a static PET image is enough. But is the same for research? We'll see. So disadvantages, the interpretation, again, the interpretation is not validated for several PET tracers. So the interpretation of higher is, this is the mean, okay. We have some match with results in the literature for to interpret the FDG, for instance, static images, but for several tracers we don't have. We still are to deal with which is the best static time windows to acquire the information. So in true, we can also, in order to deal with different type of protocols, so the injection, there's a bolus injection means that, you know, for instance, in my center we use 20 seconds, in our center they use 30 seconds, in our center 40 seconds, even we deal with the same tracer, for instance. So in order to standardize also this, we can, again, to have not to use SUV, but SUVr. So we use a reference, an additional normalization of the images using a reference region. Also, this is a critical point because this reference region is supposed to be a region non-affected by the pathology, okay? So you've compare healthy and so pathological Alzheimer's diseases, so for instance, you have to be sure that the reference region is not affected by the pathology. Otherwise, you have a bias inside your data. And or is a referent with a neurotransmitters is a referent assumed to be without specific binding, without specific receptors, without the GABA systems, for instance, for GABA studies and so on. So when you have SUVr, first you have to ask which reference was used, okay? Okay, now we have a for instance, you have this image is obtained by using RAW15. RAW15 is used for GABAergic, for investigate the GABA receptor system. And the interpretation. Higher is value, higher the receptor density is correct. Higher the value, stronger is the binding with the GABA. Or higher the value, higher is the volume occupied by the tracer. Which one is the right answer? Which one is the right interpretation? We have to go inside the literature. We have to understand what we have, which frame I have to have the right answer. So it seems to be really simple to have a static PET image, but the interpretation of a static PET image is based on several assumptions that we have to first address by using different, looking at the literature and what? First, to look in the dynamic process. Okay. How is possible? Of course, using dynamic PET imaging. In this case, again, I inject the dose. I follow all the behavior of a tracer. This is a leucine, in this case, so synaptic synthesis, not density, synthesis. And the time is, I follow the process over 90 minutes, taking different type of pictures, several 3D images. And the combination of them is, this is of course, is a continuous line. I have just only a sample of this continuous lines. In any case, this table is sufficient to follow all the behavior of a tracer inside the brain in this case. Okay, fine. In any case, each one in kilobecquerel per millimeter. So how is the most powerful? Why dynamic imaging is more powerful, even if more demanding in comparison to the static? Okay, to demonstrate this, to convince you that dynamics provide us a little bit better, in identification of a PET tracer, we need the dynamic data. We need some information coming from blood samples. In true, with the new scanners, we are trying now, there is now the possibility to derive some, instead to use blood sample, we can use directly what we call IDIF, image derived function, directly from the images. Now it's possible because, again, we have a new scanner, new possibility, new resolution, so on. So we need the true information, and we need the model and to characterize the system. Okay. So the model is not really so complex to understand. For instance, this is an example. We have a PET tracer, like row 15, for instance. And this is just a picture of a cartoon, you know, to think, to visualize what is happening. So I inject the tracer. The tracer is this one. These are the black circles that you see here. Is coming. This is a voxel. This box is a voxel, supposed to be a voxel. And there is a capillary. So the tracer is coming from outside inside and going about. And so coming inside the voxel from the capillary. Is crossing the BBB and entering the tissue. Entering the tissue and going the synaptic cleft here. And here it can found both specific receptors, specific, for instance, GABA, if it's row 15, or if we use WAY, serotonergic receptors, or unspecific, something else. Okay, this is exactly the same. Okay, how to translate this in a model? First, we use a scheme. Scheme could be this. We can just do a different type of instead to use this one with this. So there is the tracer in the plasma. It's possible if the tracer cross the BBB, enter in the tissue. There is a synaptic cleft where you can find the tracer. And here it can bind to the specific binding sites or non-specific binding receptors. Okay. And normally this is association, dissociation, rate constants to describe this binding process. Okay, this is reasonable, I think, a schematic vision of this, what is happening inside the physiology. And this is really easy to translate in a model. There are several types of models that we can use. In PET, we use normally compartmental model. So each one of this piece of a scheme is translated in one compartment. Okay, and this is the model. You cannot see, but we have also differential equations. Every compartment is described by one first differential equation. So, but in any case, we don't want to go inside the math, but to understand what we can derive from this type of information, from this type of processes. Okay, so we put some parameters describing the velocity of the process. Here, this one is K1. Remember that there is a BBB, the K3, 4, 5, 6, and so on. 6 different parameters. For each parameter, I have a unit of measurement. In particular, K1 is a very interesting parameter for me because K1 is a combination of 2 different information. The tracer is reaching the tissue with the velocity of the blood flow after it's crossing the BBB. K1 is the combination. There is a mathematical formulation. There is a combination of both CBF and BBB, okay, permeability of BBB. Okay, so it's an interest parameter because we can investigate the mismatch, for instance, of some substance that we want due to the pathology. In particular, it could be that there is an impairment of the blood flow. Okay, we see a difference K1, but also we can have an impairment of a BBB, and again, the K1 is, take into account for this impairment. And okay, this is a K1. A K2 is just only quantifies the rate of the exflux of a tracer from a tissue. Interesting, but not really so. And K3, K4, K5, K6 are related to the specific binding and the unspecific binding, okay? Again, unit of measurement in this case is a milliliter, centimeter, or the volumes, and over the minutes. And here is just only the rate constant, velocities. Okay, this one. So in truth, the model is a little bit more complex. So we can reduce the model because these actions, these two processes are very, very fast. Okay, the tracer is not, you are not on my binding side, we are not on my binding side, we are not on my binding side. So the process is very, very fast, so we can collapse the two compartments, and this is what is now we use, is the gold standard model that is used to quantify PET tracer in dealing with receptor systems. So again, CP is telling us that we need this information. It's coming from blood samples or from images itself. And these are the two compartments. So the second one is the specific receptors space, and the first one is the combination between the synaptic cleft and unspecific receptors. So now we have just only 4 parameters, and we already have seen the interpretation of K1. Speaking about K3, K4, this we have here is a reaction. There is a binding process, and the reaction is normally, is a chemical reaction. And the chemical reaction, normally, if you go inside the books, is the rates are represented by K on and K off. Okay, K3 is not directly a K on, but is a K on multiplied by the Bmax. Bmax is the concentration of receptors. So K3 is a combination of two information, the velocity of this process on, of the binding, multiplied by the concentration of the receptors. Really the concentration of the receptors in that voxel. And in vitro is exactly the concentration of receptors. In vivo, there is a little bit different interpretation, in case the values are really, really close. The concentration is unoccupied or available receptors, but more or less the meaning is exactly the same. K4 is directly the K off. What we can derive, which type of information we can derive? Of course, we can estimate for each voxel K1, K2, K3, K4, but also we can derive Bmax. This means that we can translate the dynamic information in a PET map. Each voxel is telling us the Bmax. Okay, the concentration, receptor concentration. Or I skipped this one, that is not related more to tracer than to the receptor system. VND, not displaceable, is the volume occupied by the tracer in the first compartment. Do you want this? Okay, do you want a PET map at each voxel is representing a VND? Could be yes, because due to the pathology, it could be really interesting. I don't know. But this VND for us means the volume occupied by the tracer in this first compartment. So VT total is the volume occupied here and here by the tracer. So if you have a PET map, VND is different from to have a PET map representing the VT. The physiological information inside are different. And I can also provide a map with of a BP, binding potential. The binding potential is the ratio between K3 and K4 is strongly related to the concentration of a receptor sphere, even if it's not exactly Bmax. So is related, strongly related to the concentration. So, and normally, if you go inside the literature, you can go inside the data sets, you have PET maps, or normally it's written, I use PET maps. And which type of PET maps? PET SUV, SUVr, and Bmax, VND. Are you sure which type of information you have in your hand? And are you sure that you are not, mixing for instance, Bmax from row 15 to with a BP from, or VT from, for instance, the DOPA, with some V and D from another tracer, what I have is exactly the pool of PET maps that I have are telling us exactly the same information or not, and which information I have. This is something that you have to have. So I skip from receptor. The last one is glucose metabolism with PET. So now there is a different. First we had receptor studies. Now we have glucose, so metabolic studies. And in this case, we don't use directly glucose, but FDG, because glucose itself has a much really complex model, several compartments, several parameters. Is too much for us to deal with. There is some mathematical issues associated. So normally we use FDG. FDG follows just only the first step of a glycolytic pathway. And of course, making simpler the model that is used. So again, we can have SUV or SUVr with using FDG, or we can have also some dynamic study. So SUV, normally the interpretation is higher is the value, higher is the glucose consumption there. And I wanted to also stress the additional information that we can derive from dynamic acquisition. So the meaning of a model is a little bit different, of course, because we are dealing with glucose, not with receptors. Remember the BBB, the BBB for glucose sensitive to the presence of GLUT1 and GLUT3 that are the transporters of glucose from the blood inside the tissue. So K1 in this case is, again, remember, it remains related to the blood flow and the BBB, the permeability of the BBB. So we can investigate if there is an impairment of the GLUT1 and GLUT3 present in the BBB, if we have a K1. K3 now is quantifies the intracellular glucose phosphorylation rate, the first step of glycolysis. Okay, and we can also derive a macro parameter that is called Ki, that is a combination of three parameters that is called fractional uptake ratio. This macro parameter is sensitive to a modification of a K1, a modification of a K2, a modification of a K3, okay? So SUV in FDG is strongly related to the Ki, okay? It's not related to the K1, it's not related to K3. K3 is phosphorylation, is FDG, is glucose that is transforming in the first step of a glycolytic step, glycolytic change, okay? So these are maps. So I can identify my model in each voxel and can provide you with 4 different, of course, static images. The first one is a K1. What is happening inside our brain? This is healthy. It's a combination of the group average of 54 subjects coming from Washington University, provided us by Washington University. It's healthy. They are healthy. So this is K1. Ki, SUVr, reference in this case is a global reference, and K3. K2 is it's very uniform, okay. Okay, well, but the picture is a little bit different. You give a look, for instance, of a Ki here, K1, and K3 in the occipital area. And also give a look to what's happening in the rear part in comparison to the frontal part. And the interpretation, because here the K3, the phosphorylation rate, is telling us, giving us a little bit different picture from the Ki for the overall glucose consumption. Ki is a combination of a K1, blood flow, BBB, K3, phosphorylation. This is just only phosphorylation, K3. A K1 is related to blood flow and BBB. Okay. Again, I use I use exactly the same tracer, FDG. I have 4 different vision information of the same process. So now what we wanted to use, of course, depends on the issue, of the aim of the project, the aim of the pathology, whatever I have in mind. So, but you have a richer information, of course, very detailed information because it's physiological based information. And this is something that, you know, now you can use all of them, but the meaning is different, and the vision of a glucose, in this case, is different based on the different parameters, of course, because it's different steps. Okay, I want to thank you for your attention, and, you know, just, you know, sorry if I was not clear enough, but, you know, questions? No question? One question? Something?
I think it's one there.
Okay.
Hello. Thank you for the great lecture. I had a question to calculate SUVr.
Yeah.
Do we need to know the tracer injected dose?
SUVr, you need, for SUV, you need the dose. And also the weight of the kilogram of a person. And with R means that you use also a reference region. I cannot say you which reference region because it depends on the tracer and depends on the pathology.
Because the tracer injected dose would be cancelled out when you divide the SUV from 2 different regions.
Yeah.
So I was wondering whether we need to have it because, for example, when you use like a lot of datasets available in the publicly available databases, a lot of them don't have that information. Yeah,
you can use directly the reference region to provide SUVr because, of course, there is the dose and the same. And it's normally the steps are that one. Okay. And the last one is really more to clean the differences in the protocol.
I see. Okay. And another question that I had, in the last images that you show, it seemed to me that the spatial resolution of K1, Ki, and K3 is different from SUVr. Is that correct?
Of course, we can, normally we have SUVr, we use the clinical images. And for the dynamic images, we use a different type of algorithm to reconstruct the images. This is something that is used in PET. We have several different type of algorithms. Fast algorithms are used for clinical purposes, but normally there is the smoothing there. There is a smoothing processes that is evident for instance, when you open SUV or SUVr image, normally there is a smooth process there. And in our case, so dealing with dynamic images, we prefer to conserve the original sample voxel size, so we just move it to a different algorithm without smoothing. And this at the end it has an impact of the quality, of course.
Thank you.
Hi, I just wanted, so for the SUVr, which R do you tend to use, or does it matter? I know it has to be one of the ones that's not interesting, but there's still some choices, I guess, right?
For FDG, in this case, due to the fact that we have a normal subject, we use the average signal. The average time activity curve. This was the reference. That is normally due for FDG in...
average across the whole brain?
Yeah, whole brain. Okay. Whole brain. Whole brain. And whole brain, gray and white matter.
Yeah,
okay. Not CSF, of course, and not skull, whatever, so but whole brain.
And I had another question, which is, so like in fMRI, you tend to smooth the data when you do this kind of analysis. Is that also done in this domain? Like with the kernel?
We don't, I don't smooth.
Okay.
I prefer to, avoid this one because due to the partial volume, of course, and so on. Yes.
Okay. Thank you.
Thank you.
I have a question, actually. So you touched a bit the point about, putting together images quantified in different ways. So I have a very practical question. Like I used neuromaps, and in neuromaps we have maps based on Bmax, BPND, SUVr, and VT, even for the same tracer. So the question is like, do you have a practical recommendation of what to do? Like try, is there some measures we should prefer over others? Should we just, consider one, or if we have let's say mismatching results, should we go for one, or what do you recommend?
So from ideal point of view, the best is BP and Bmax. Okay. From practical point of view, of course, we know that the most reliable measure that we have is the VT, more stable, more easy to obtain, also because we can use a Logan and what's on. And Bmax is very challenging because it requires also an additional effort from an experimental point of view. And the BP depends on which type of, if you are really good in the nonlinear estimation, or if you have a reference region approach, reference region approach. I mean, it's different stuff. We can quantify with different type of approaches the microparameters. And if you use a reference region, again, there is a problem. What a reference region is, in fact. Is really devoid of trace, devoid of receptors. We know the cerebellum, in any case, even few, in any case, it has receptors. So stuff like this. So theoretical point of view is something, practical is something else. All is clear. So we can switch to Martin. Martin is assistant professor in the Department of Computer Science at the University of Copenhagen. He has done extensive work on understanding analytical variability in PET processing, which has led the PET field toward more open and reproducible research practices. Furthermore, he has led international data sharing and guidelines initiatives encouraging the field to openly share PET data, because open data is very expensive to acquire, very expensive. I know. Because, fMRI, I don't know, is something 200 euros, something like this. I don't know. And PET, oh, really? In any case, PET, I don't know. I know FDG is cheap, but with some tracer could be 3,000, 5,000, 6,000 euros for subject, okay. And to increase sample size to attain more robust and generalized research results. Sorry for mistakes, and the floor is yours.